So we have now made our way to talking about the scientific method. So the scientific method is a series of logical steps that are used for testing a possible answer to a question. So these steps are a guide. They are not a rigid sequence. Steps may be followed in a different order. They could be skipped, or some steps may end up even being repeated. So it's very important to know that this scientific method is an oversimplified way of doing science. Remember, science is so complex, so there's no one right way to do science. So there's not just a single scientific method. Many scientific discoveries have been made by accident. So one of the theories or ideas of how dinosaurs went extinct, and even the discovering the structure of a DNA molecule were all discovered by accident. And so science isn't done in just this one way. So when we look at the scientific method, we're going to look at all the different steps to it. So the first step says to make observations. So an observation is anything that is detected with one or more of your senses. So to remember, as a reminder, your senses include your sense of sight, things you see, things you hear, your sense of touch, how things feel, is it bumpy or smooth, the way things smell, and how they taste. Your second step is to ask a question based on observations. And the third step is to form a hypothesis. And a hypothesis is a potential, testable answer to a scientific question. When we say testable, it means that if the hypothesis is false, it is possible to find evidence showing that it is false. We can't just test something that will be true 100% of the time. When we make a hypothesis, it usually requires the scientist to do some research. They need to find out if and what other researchers have learned about the topic. So once you have written a hypothesis, you can perform your experiment. And the experiment is a controlled scientific test that often takes place in a lab. Notice it says often and not that it has to. So when we do experiments, there are a couple different parts. The first part of an experiment is the independent variable. This is a thing in the experiment that is manipulated or changed. Your dependent variable will depend on the independent variable. This is the results of the experiment. And lastly, we have the controls. The controls are other factors that do not affect the dependent variable. These are kept constant, so they will not affect the results of the experiment. So if we look at this example here, this maybe will help give you some clarity. Josiah wants to know if adding a fertilizer to his tomato plants will cause the tomatoes to be bigger. Josiah takes 4 pots and adds the same amount of soil to each pot. The soil all came from the same bag of potting soil purchased at Menards. In each pot of soil, Josiah plants 3 seeds of his famous Roma tomato variety. Each pot is put in the same place and will receive the same amount of sunlight. He waters the pots the same each day. In the first pot, he does not add any fertilizer. In pot number 2, he adds fertilizer every 3 days. In pot 3, he adds fertilizer every 2 days. And in the fourth pot, he adds fertilizer every day. So in this experiment, I'm wondering what the independent variable is. So remember, the independent variable is the thing that changes. And so what in this experiment changed? So when you're reading through, anytime it says the same, the same place, the same amount, the same each day. That's not changing because it's staying the same. So if you look at this last bullet, it says that he does not add any fertilizer. Then he adds fertilizer every 3 days. Then he adds fertilizer every 2. And then lastly, fertilizer is added every day. So the thing that is changing is the amount of fertilizer each plant is receiving. So then we have to look for the dependent variable. And remember, the dependent variable is your end result. What is he measuring? And so that's up way back at the first bullet already. He is adding fertilizer to his tomato plants because he will cause the tomatoes to be bigger. So the dependent variable is going to be the size of the tomatoes. And then lastly, it's looking for controls or things that are staying the same. So that would be anytime you see the same written up above. So he adds the same soil, the amount of soil, the number of seeds, because he's only planting 3 seeds. It's the same variety of seeds, same amount of sunlight, amount of water. All of those things are controls. You will almost always have multiple controls and only one independent variable. So your 5th step of the scientific method is to analyze the result and draw conclusions. You are looking if your evidence supports or does not support the hypothesis. In science, we never say that a hypothesis was proven to be true, because that indicates that it can never be proven false. And that's just not the case. Scientists are always prepared to have a hypothesis not be supported. Lastly, the 6th step of the scientific method is to communicate the results. So when you communicate your results, many scientists will present their findings at a scientific conference or publish articles in scientific journals. And when communicating the results, the scientists will describe their hypothesis, how it was tested, and the results of the experiment. Doing this allows other scientists to repeat the investigation. And replication is such an important part of science. And this is when many different scientists conduct similar experiments. If each of those scientists get similar results, it makes the results more reliable if they can be repeated. So we like this, or scientists like it when their experiments are replicated and they get the same results as the original scientists. It makes the results more valid.